Yeah. Today I will talk about changes of the nuclear landscape during pre-implantation development of fertilized and cloned bovine embryos. And my talk will consist of 3 parts. It's an introduction about chromosome territories and chromatin domains. Then a talk about the main subject of bovine pre-implantation development, and then I want to give you a functional model of nuclear organization and some future perspectives. Gunther Blobel, in a seminal paper in 1985, said the genome of a higher eukaryotic organism is organized into a number of distinct 3-dimensional structures, each characteristic for a given differentiated state. These discrete 3D structures are envisioned to develop in a hierarchical and largely irreversible manner from an omnipotent 3D structure of the zygotic genome. And it's my opinion that understanding the functional nuclear organization in space and time depends on understanding its structure-function interactions at all levels of organization, from molecules to the entire system. Now, what is clear is that we have chromatin fibers with DNA running around the nucleosomes, and that we have chromosome territories, but there is a big gap of knowledge how these structures are interacted. Now, based on the evidence of territories, this cartoon argues that within a territory you have intermingling chromatin fibers. In 2000 and 2001, we proposed this model that a nucleus is organized in territories which are built up from chromatin domains in the range of 1 megabase pair, and that there is an interchromatin compartment starting at the nuclear pores, pervading the nuclear interior, including the territories, and that macromolecular complexes for transcription, splicing, DNA replication, repair should be enriched within this compartment. Now we know that we can visualize in life replication foci, taking cyanine 3, DUTP, for example, for labeling, and H2B, GFP for visualizing the entire chromatin. And when you do that in one cell cycle here during early S phase, and you follow such a cell through several cycles, you see the separation of chromosome territories, and you see that these domains stay through time. Now, at this time, we could only speculate how an individual 1 megabase chromatin domain might be organized, maybe in smaller subdomains of, say, 100 kb or so. It was impossible to study this question by microscopic methods. Now, super resolution fluorescence microscopy earned the Nobel Prize to Eric Betzig, Stefan Hell, and William Mörner. And in the scientific background, also structured illumination microscopy is mentioned, and I will talk to you about this method, and it has been independently invented by Rainer Heinzmann and my brother Christoph CREMER and the late Mats Gustafsson. Now, you have in a 3D sim study more than 1,000 images per nucleus, and you need to do a reconstruction and an image analysis. But this method gives you an 8-fold increased resolution in 3D compared to conventional confocal light microscopy. Here is just an example of when we make a pulse with EDU for 10 minutes in a nucleus and count in the entire nucleus these domains. We see here that we in an early S phase, we see some 6,000 of these domains, and arguably the number of domains may be such that you have something of about 500 kb or so, or between 1 megabase. And I think it's interesting to argue, what does that mean, chromatin domain based on replication foci, the argument of Giorgio Bernardi for isocores, and topologically associated domains. Are they basic units of higher order chromatin arrangements? This image shows you the advantage of a section with structural illumination microscopy compared with a confocal section, and you see much better the clustering of the chromatin, and you see here evidence for splicing speckles in red in between in these what we call interchromatin compartment lacunas. It's also, there's evidence that the channel system really goes to the nuclear pores, penetrating the peripheral layer of heterochromatin and being kept here in blue by here when we have immunocytochemistry of a nucleoporine. Now you can go through an entire nucleus, and in green you see lacunas from the interchromatin compartment. You can also do the following, that you use express an histone H4 tagged with a photoactivatable GFP, and you can with a microbeam then of 440 nanometer illuminate stripes or whatever you want to do here. You illuminate a stripe and you activate the photoactivatable GFP, and you can now see through time what happens, how dynamic that is. And you see there is locally constrained movements. We are now here 7, 8, 9, 10 minutes, and it goes on, and we have followed that over 10 hours. And you see that the stripe is broadening somewhat, but there is no major movements of some territories away from this region. So we are talking about locally constrained movements of chromatin domains, which can come closer together, a little bit farther away from each other. The same approach can also show you that there are indeed cells, embryonic stem cells, where within 40 minutes, when you make a stripe pattern, you have major changes of the pattern, while here in a fibroblast nucleus, like a couch potato, within that frame, nearly nothing happens. So yes, nuclei can change the shape, and it can have effects on the organization, on the arrangement of chromatin inside. I come to part 2, reprogramming of fibroblast nuclei in cloned bovine embryos. And this study is just in press, and a collaboration of my group with the chair of molecular and animal breeding and biotechnology. Now, when you consider the development of a bovine zygote, it's now known that at the 8-cell stage, there is major genome activation, and it's a critical period. You got a lot of things from your mom, but that's going to be depleted, and you have to do your own business, start with your genes, and go ahead. And I was interested to see what happens with the nuclear architecture during that period. Now here you see SIM sections of nuclei, which we see in particular around the 8-cell stage when they pass through major genome activation. Chromatin is clustered at the periphery, and there is a large central lacuna. And thereafter, nuclei get smaller, and the chromatin is pervading the entire interior space. And we call this type of nucleus embryonic nucleus with peripheral localization ENP, and after major genome activation, this we call an embryonic nucleus with a conventional nuclear architecture. This was really a surprising kind of architecture. Now, at this stage, there is starting nucleolus formation, and the internal lacuna is filled with splicing speckles. And after major genome activation, we have big nucleoli here marked by B23, and here you see the splicing speckles throughout the nucleus. So that is going to happen. These ENP nuclei cells are at different stages of the cell cycle. When we do pulses with EDU, we see that some of the nuclei in the same embryo are heavily labeled. They are in S phase. In other nuclei, you may see that H3S10 phosphorylated is here be seen. So this is going into mitosis. This is during S phase. So it's not just a synchronization. And we have looked for the topography of RNA polymerase II and H3K4 3-methylated as a marker of transcriptionally competent chromatin together with DAPI stained DNA. And when you look here on these little boxes, you see that chromatin DAPI is clustered. And you see here in green H3K4 3-methylated. The black dots show you co-localization with DAPI stained pixels, and the green pixels of H3K4 3-methylated. Here you see RNA pol2, and you can see some association of this transcription competent chromatin with RNA pol clusters or machineries. This nucleus here after major genome activation looks very different, but the local organization of the chromatin, which we can see, is very similar. Now, when we look on the nuclear volumes, we see a highly significant change of the nuclear volume after major genome activation. The nuclei get indeed smaller, and we see now the change from this ENP architecture to what we call the ENC, the normal architecture. A switch to cloned embryos, and Eckhard Wolff's group has developed a fibroblast, fetal fibroblast line, which expresses a reporter gene called GOF, and that is integrated in the chromosome 13. And it combines regulatory sequences from OCT4 with coding sequences for EGFP. And interestingly, when they make biosomatic cell nuclear transfer embryos, the blastomeres start to get green when the embryo passes through the 8-cell stage. Very similar as major genome activation in vitro fertilized embryos. we see then that we have here the flat ellipsoidal fibroblast nuclei, and they are forced to make very similar changes of the nuclear architecture, getting big with chromatin at the periphery. These individual clusters form individual chromosome territories, and thereafter the nuclei get smaller and we have the conventional nuclear architecture. Here you see an example in a fibroblast nucleus, flat ellipsoidal nucleus. Here you see the clustering of H3K9 3-methylated versus H3K4 3-methylated H3K9 for a marker for silent chromatin. So it's very different distributed. When you look here in detail on the distribution of H3K4 3-methylene RNA polymerase II, this is enriched in the periphery of interchromatin compartment lacunas. And here is an example now from a cloned 8-cell embryo where you see H3K4 3-methylated together with some RNA Pol2. And I will show you a little bit later how we have quantitated that topography in such nuclei. This is a nucleus of somewhat a later stage, and you see the nucleus is smaller, and you see again nicely the enrichment of H3K4 3-methylated in the periphery of interchromatin compartment lacunas together with RNA Pol2. Now when we look on what happens with the volumes of nuclei in cloned embryos, this is the volume of fibroblast nuclei. Then is the enormous increase in size of the nuclei at stage 2 of cloned embryos, and thereafter the volume decreases again. But when you measure a roundness factor, you see that here the fibroblast nucleus rather flat, a low roundness factor, it increases, but the roundness factor stays. So these nuclei became round and they stay round. Also they decrease in size. Now here you see a heat map of intensity classes 1 to 7 of DAPI stained DNA, both in vitro fertilized bovine embryos and in cloned bovine embryos. And that is the kind of changes during pre-implantation development. And that is what we have used for quantitation of histone marks and RNA Pol2. Now, here you see with our classification that at day 7, 8, with 7, 8 cells in vitro fertilized embryos, most of the nuclei show this ENP phenotype with the peripheral chromatin and a major lacuna in the interior. And then this changes, and in embryos with 18 or more cells, most of the nuclei show the conventional architecture. In cloned embryos, we have also a large fraction, nearly 50%, which shows this ENP phenotype at the 8-cell stage. And we have also studied blastocysts, where basically all the nuclei show the conventional nuclear architecture. Now, 5 minutes. Now you see that when you do these heat maps, you can count the pixels in each sections which belong to the different classes. And when you do so, you can ask the question, are your, say, pixels, immunopositive pixels from RNA polymerase II randomly distributed so that each of these fractions has the according fraction of RNA pol or histone marks, or does it deviate from a random distribution? And our study very clearly shows for each of these pictures is based on the evaluation of some 10 nuclei, that there is a highly significant enrichment of H3K4 methylated and RNA pol II in classes 4 and 5. There is, in contrast, a significant enrichment of H3K9 methylated in class 7, the most densely packed DNA. And there is an underrepresentation in classes 1, 2, 3, which belong to the interchromatin compartment and the more decondensed periphery of the chromatin domains. And this finding we see in vitro fertilized embryos, in cloned embryos, and in fetal fibroblast nuclei. Now, briefly, at the end, a functional model of nuclear organization as we see it now and some future perspectives. We argue that interchromatin channels start at nuclear pores and pervade the nuclear interior, and that small loops may extend from the periphery of chromatin domain clusters in the interior of this interchromatin channel system. And this here shown in green is what we call the active nuclear compartment in 3D, paralleled by the silent compartment, which is a clustered interior of the chromatin domains. And like a natural landscape, you can see here with these little streams where something is going from the shore into the interior. Now, what one would like to do is making quantitative 3D and 4D maps that precisely communicate structural information of nuclei in space and time at the nanometer scale. And the way to do this is a kind of molecular pointillism, and Eric Betzig received his Nobel for the invention of this approach. When you have such a painting and you have very little dots, you see nothing, more and more dots with better and better resolution, and you see the whole picture. When you use blinking events of GFP or other fluorophores in a way that each blinking fluorophore is separated statistically further than the Abbe limit, then you can pinpoint the localization of each blinking event very precisely. And you can accumulate these blinking events. And here I show you one of the recent images made in my brother Christoph's lab. You can count millions of individual, each little point is an individual blinking event here of a DAPI staining fluorophore called fibrant, and you start to see the channel system. You can do millions of them, and you go down from a resolution of some, with structural illumination microscopy, of some 100 nanometer lateral, 300 nanometer axial. I think that's possible to go now to 30 nanometer, And then you are at the right resolution really to study the organization of individual chromatin domains. So this is my lab, and I should thank in particular my wife Marion and Jens Popken, who has performed all the experiments I showed from bovine embryos. I have a collaboration from the kindergarten with my brother Christoph, and this is the group of Eckhard Wolf, and Volker Schmidt supported us with the statistics. Thank you.